Okay, next we have Niki Armstrong, who's FAST genetic counselor, who's going to be talking to us about cracking the code, understanding UBE3A mutations.
Okay, so we've talked already a lot today about the UBE3A gene making the UBE3A protein, but there's one key piece that I wanted to cover when we're thinking about mutations, and that's how the gene codes for the protein. So in the picture here, you can see that the gene is made up of chemicals that we call nucleotides. So those are the A, C, T's, and G's shown there. And the body reads those nucleotides 3 at a time. And for every 3 of those nucleotides, it puts on 1 piece of a protein. So 3 chemicals codes for 1 amino acid or 1 piece of the protein. The cells read the gene, make the string of amino acids from the code of the gene, and that string of amino acids goes through complex folding and processing to make the functional UBE3A protein. And that's shown there as the green blob. So when we have a mutation in the UBE3A gene, there's a change within the code, within those chemicals that make up the gene, that makes the resulting protein either absent or non-functional or less functional. And that change could be just 1 nucleotide that's affected. It could be 50 or a couple of 100 nucleotides affected. But for it to be mutation AS, it has to be completely within the UBE3A gene. And it's really important to understand that there are hundreds or maybe even thousands of different mutations within the UBE3A gene, and they can all affect the protein differently. So we've already talked a lot today about what the UBE3A protein actually does. One key role, of course, is that flagging that we talked about, so adding ubiquitin to other proteins to help the cells know it's time to break them down. In order to do the jobs that the UBE3A protein is supposed to do, it has a number of different domains or sections of the protein that interact with the other proteins. So we saw some of that in earlier talks today. And you can see some of the 3-dimensional structure that's shown there that looks like the curlicues. Those are some of the different portions that interact with other proteins. And those are all really important. If we have disruptions in the way that some of those structures are made, they're not able to bond to or to associate with the other proteins that they need to. And there are parts of the UBE3A protein that are completely necessary for the function. One key role there, if you can see that teeny tiny little red star, that spot at 820, that's the spot where ubiquitin actually bonds or attaches. So if that spot has a problem with it, the UBE3A isn't going to be able to do any of its adding the tags onto things. So we know that mutation AS is really variable. It can affect each person living with mutation AS differently, different degree of symptoms and severities, and a lot of that has to do with the fact that each mutation can affect the protein differently. So we set out to better characterize the mutations. We started looking within the Global Angelman Syndrome Registry, and we had 61 participants there with the mutation genotype. But then we wanted to broaden that. And so what we did was actually look in a number of other data sets, and we ended up with a total of 279 different UBE3A mutations that we classified and characterized based on what they do to the gene itself and then what they do to the protein. And for more details about this, I'm happy to go through it. It's all on my poster in the poster session. So in trying to understand the variability in the mutation AS, there's sort of 2 key pieces. One is where in the protein are the mutations. So what we're seeing here on the bottom is the protein again with the different domains. And then what we're seeing on the top is a mapping of the mutation at the amino acid that it occurred at. And the key pieces here are, 1, mutations are throughout the entire protein, right? You can see little blue lines throughout the entire protein. But then if you look towards the end of the protein, so around the 800s there, where that key spot is where ubiquitin binds, you can see more blue there and higher blue lines. That means there are more mutations affecting that part of the protein. That part of the protein is very important, and so that's what we see there. The second piece that causes variability in mutation AS is that each mutation can affect the protein differently. We have some mutations that result in no UBE3A protein at all, some mutations that result in a UBE3A protein that's just missing an important piece, like missing the ubiquitin piece. And then we have some mutations that result in a UBE3A protein that has a completely abnormal structure. And so it's sort of hard to predict what it can do and what it can't do. And you can understand that these different kinds of mutations and different UBE3A proteins might interact differently, right? And cause maybe a different kind of symptoms. And also, we might need to think about therapies differently depending on the effect of the protein. So, bottom line is, mutations are really complicated. We can classify them based on how they affect the gene. But more importantly, really, is understanding how they affect the protein. And what we really need to do are collect and map and model as many mutations as possible. So especially if you have a loved one living with mutation AS, please join the Global Angelman Syndrome Registry and update your account with your registry or with your genetic test report so that we can make sure we've got a broad base of mutations. Thank you.
That's so important, and it's critical as we think about inclusion criteria into clinical trials. And I want everyone to just accost Niki throughout the rest of the weekend if you have a child with a mutation, so she can talk to you and really gain the knowledge of your child, and you can share that genetic information so she can help navigate this. So I think it's really, really important that we best understand mutations in Angelman syndrome.